we're extremely grateful for your leadership and your support, especially during these challenging times. Last week, I actually celebrated 15 years representing Weizmann Canada. And as you've heard me say many times, I still feel the same level of excitement each and every day as I did on that day so many years ago, even more so. Much as the past 15 years has been about learning and sharing and included many moments of pride and gratitude, I have never been more proud of the Weizmann Institute and our scientists as I am during this time. And never have I been more convinced in the value of basic science combined with the multidisciplinary approach that is the very DNA of this institute. From more efficient testing to predicting outbreaks, to smell tests you can take at home, to novel ways of developing antibodies, vaccines, and even an exit strategy, the Weizmann way of approaching a problem is unique and impactful. I'm proud and grateful to be able to share science and hope during these challenging times. And these initiatives are all made possible through the generosity of our philanthropic partners all over the world. And we're extremely grateful. Speaking of being grateful, I thank Professor Alon Chen for his leadership and for taking the time to be with us today. And a special thanks to our national chair, Jeff Cohn, who will introduce Alon. Thank you, Jeff. Thank you, Alon. I will now go on.
Susan.
Thank you, Susan. Welcome, everybody. I hope everyone is doing well and managing to keep good spirits during these obviously extremely challenging times. So Alon is actually a neuroscientist who has made major strides in elucidating the brain's activity in stress and anxiety, and is the president of the Weizmann Institute of Science. So that could result in multiple jobs when you're management and still continue your professional activities. Previously, Alon was the head of the Department of Neurobiology and director of the Max Planck Institute of Psychiatry in Munich, Germany. He leads 4 donor-funded multidisciplinary centers in brain research and is also a committed proponent of science education and science literacy. I've had the pleasure of hearing Alon speak on many occasions, and I have to tell you that it's always extremely interesting, but more importantly, always extremely relevant because you can take a lot of what Alon says and you can actually personalize it because he speaks in many cases about how we manage stress and what stress does to us and the consequences of stress. And today, Alon will be speaking to us about some of the many projects on the campus that are making an impact in solving the COVID-19 crisis, as well as how people cope with stressful situations, which unfortunately are a bit too prevalent these days. So we're honored to have you here, Alon, and thank you very much. And we look forward to hearing what you have to say.
Thank you very much, Jeff. Thank you very much, Susan. And of course, thank you all for joining us. Thank you for the ongoing support and generosity. And as Susan mentioned, this is so important, specifically, especially in these very challenging days. So what I will try to do today, around half an hour, I will start by giving you some overview about the activities that are going on regarding the Corona at the Weizmann. Of course, we have more than 50 projects, so I will just mention some of them today. And then in the second half, I will tell you a little bit from my own expertise, as Jeff mentioned, about stress. I think it's always good to understand what happened in your brain and body, what those changes that are very common these days due to the uncertainty and other different type of stressors. I think when we understand better what is causing those feelings, we can cope with them much, much better. Of course, we will have plenty of time for questions. I think it's not a very big group, so Susan can just forward or just tell the question, announce the question loudly, and then be happy to discuss either Weizmann and of course about stress. So the last few weeks, I think it's already almost 6 weeks since we made this major shift toward the Corona research on campus. Things really changed here. First, we have to implement new regulations that we receive from the government, and It's not trivial because the institute is currently working on 30% of its manpower, and these are government regulation that we have to obey. So it's working day and night in order to try and keep this institute functioning. And around 4 weeks ago, we received a request. I actually attended a meeting with the Prime Minister Netanyahu and other leading bodies, and we were asked to try to establish labs at the Weizmann that will conduct tests for the coronavirus on campus. As you all know, we are a research institute. We don't have clinical labs for doing tests like this. Although we have the best lab in the country and we have the best equipment in Israel, definitely, we had to adapt those labs in order to stand in the clinical criteria and also, of course, get the certificates and get the training, get the right protocol to do those tests. And we started a very big project in our national center, Nancy and Grant, the Stephen Nancy Grant Center for Personalized Medicine, the INCPM, which has been transformed to be a huge center for corona testing. We use all the robots and the sophisticated PCR machines we have there. And in the last week, we already start running tests for corona. I will give a little bit more information about exactly what type of test. There is a lot of issues that we are facing every day, but you should know, and probably you know this from the news. I don't know what the situation currently is in Canada, but Israel is already in a position that is moving from blocking the spread of the virus. So the strategy now is less about holding the spread and being worried very much about the severe patient. And we already start moving into what we call exit strategy. So what the Weizmann is really engaging now in the test field is mainly checking not only the sick people, but rather checking the population, the overpopulation. And we just finished a big screen for all the policemen in the country, just to get a better notion about what are the number of infected people on the healthy population, which we think it's extremely important for the exit strategy of the country that hopefully will start immediately following the holiday. Actually, in 1 hour and 20 minutes, we're going to start a full lockdown in the country. So no one can actually go outside of their own city or small village. And this is due to the fact that the government is really worried about people due to the holidays, a very family holiday, as you all know, that people will not start meeting. So everything is on a lockdown, although the situation in the country, if you compare it to any other country in Europe, definitely we are in a very, very good situation. And the curve is starting to flat already a few days ago. So we hope it will stay like this and we can start the exit strategy immediately following the holiday. But of course, this is the optimistic scenario. So one effort is, as I mentioned, is doing those tests, helping the country. The Weizmann invested millions already of shekels in transforming and buying the relevant equipment, buying the relevant kits. And it was a very good decision not to wait for the Minister of Health to support this. Now it's government, it takes time. Although we got the direct order from the Prime Minister, we decided to go ahead and make those purchases. And it was very smart because now everything in shortage and we have a lot of the equipment and mainly the consumables, the kits to do those type of tests. So this is one effort. But the second effort is in parallel, which also relate to the tests, is the fact that in early stages, we realized that the way the tests are done today, it's not the optimal way. It's not the most efficient way to do tens of thousands of tests in Israel. Currently in Israel, the first week of the outbreak, it was 400 tests per day. Now it's around 5,000, 6,000. For the size of Israel is good. They're aiming for 10,000. But we knew that in order to eventually go out of this situation, we will need tens of thousands of tests every day, even 100,000 and 200,000 tests per day. And this is the reason we started another initiative, which is a research initiative, and is to develop a test which is cheaper and faster than the one that is currently run all over the world. Also a PCR-based test. And this is a project led by Professor Ran Elinav and Professor Ido Amit in the Ullman building. We just the recently renovated building, we have this empty space, and in 4 days, we collected equipment from all over the campus, robots and all the real-time PCR machines, which we have many on campus, and we put them in one location, one unit. And in the last 2 weeks, Eran and Ido and many other people at the Weizmann developed a new test, which is faster. It's a test which do not require the isolation of what we call RNA. The samples are arriving on a plate, a 96 well platform plate, and the test is is cheaper and is much faster. And we already finished doing validity, the quality of the test on soldiers last week, yesterday in a full department in one of the hospitals nearby. And today there was a big thousands of soldiers in one of the Air Force base were screened in order to finish the validation. And this unit can make 20,000 tests per day, which is amazing. And we already have people approaching us from different countries. And it has been presented to the government yesterday, and it's all over the news here. And we really think we can make an impact by this. By the way, I just maybe mentioning when we're talking about the exit strategy, you should know that another option is to take samples from 10 people and pull it into a single test. So if we have 20,000, we can actually sample 200,000 people. And this, if you do the right mathematical models, we can actually, by having 500,000 people which are pooled, meaning 50,000 tests, we can actually get this country totally open based on this. So we are very excited about these efforts. So in addition to the test-related effort, we also have a lot of other activities which are aimed toward developing a new vaccine or vaccination, or also develop or isolate new molecules which will target either the proliferation or the infection of the virus. We have, I would just mention that before the corona pandemic started, we had only one lab at the Weizmann which actually worked on this similar type of viruses, lethal viruses. And this is the lab of Ron Diskin from the Department of Organic Chemistry. And this is the reason Ron was able to do a very fast shift toward the corona, the COVID-19 strain. However, currently we have 50 labs working on the coronavirus. So 49 additional labs that used to do totally different things, took their knowledge, their equipment, and just shifted toward working on the coronavirus. And some of these projects are just amazing. And maybe I will mention very briefly only some of them. You can also go to the website, and I'm sure you can. We also released some of the movies that you received recently, and we update the website on a daily basis. But maybe I will mention the amazing work done in Nir London's lab. Nir is working also from the Department of Organic Chemistry, and Nir is searching for molecules a small molecule which will bind a very specific protein, a protease, which is extremely important for the ability of the virus to penetrate our cells. And he's using computational model to screen the exact location of this protein, how the molecule can bind. And he screened more than 1,000 of these molecules, and he isolated initially 60 molecules. Now he narrowed those into 30, and they're working really on the best molecule who can bind this. This is an open source effort. It's in collaboration with other colleagues in Oxford, in the UK, and also in the US and Germany. and everything is open source without any regulation or patent. And it's amazing to see the collegiality of the people in these days. And they're already testing the molecules that Nir has developed, and Nir is leading this amazing project. Additional project added by Professor Sarel Fleischman. Sarel is expert in proteins, and as you know, antibodies are proteins. And what Sarel is doing is also shifted his lab. He worked before, by the way, on other viruses before, so he had the knowledge, and he shifted his lab now, working on COVID-19. He's also using computational models, focusing on a different type of protein in the coronavirus, and he's also developing what we call a nano antibodies, very small antibodies based on the 3D structure of the antibodies developed against the body is developing against corona, and he's also already in quite advanced stages of in his research. The last one in this domain I will mention Ron Diskin, as I mentioned before, and Ron since worked before on similar viruses such as Ebola or HIV. He's taking two different paths. One is developing against small molecule which will target the virus and prevent him from being proliferating in the cell, but his other efforts are going toward isolation of the antibodies from patients that people who were infected but now are recovered, and by isolating or determining the 3D structure of those antibodies, he can really mimic or can really see the site where this antibody is targeting the coronavirus, and then can mimic this. This also, again, a big collaboration effort. So there are multiple other groups which are working to try and find solution toward vaccination or treatment. We have other groups of scientists which are focusing on mainly using artificial intelligence tools and predicting the spread of the virus in the population. Probably the most advanced project is headed by Professor Eran Segal and Professor Benny Geiger, and they developed this questionnaire, which is already, I think, almost 4 weeks since it's been released. We have 400 to 500,000 people which fill this questionnaire every day now in Israel. It's based on symptoms. Everyone to say if you have a fever and how old are you, whether you're smoking, not smoking, and so on. And by using machine learning algorithms or artificial intelligence algorithm, Iran could predict where the virus is spreading and where the next outbreak is going to be. And he did it for the whole 2 to 3 weeks, informing the Minister of Health where the next site going to take place. It is amazingly did it, and he predict all the main sites in Israel 3 to 4 days before. And this is extremely helpful in order to monitor the spread of the virus. And now, again, it's been shared with many other countries, and it's become extremely successful. Another important effort is in science education. I should mention this, especially our educational arm of the Weizmann Institute, the Davidson Institute. And as you know, we don't have school already for a few weeks, and many kids at home. And the Davidson really in early stages decided to release this website called Stuck at Home. And they took many of the online activities and concentrated them in this site. And originally our website is in Hebrew and in Arabic, but they also, the week after, they already translated the website into English. So many of the activities are in English. And while in usual days we have thousands of entrants to the website, now we have tens of thousands of entrances to these activities, and it's amazing. It's all over the news again. And we are currently busy with translating this also into Spanish, try to really help people around the world. So I think I even longer than half of my time, so I will shift now to the other topic, but then we'll have plenty of time to ask questions about the our efforts with the corona. So I want to say something about stress. Again, we don't have too much time, but I think it's very challenging days, and we all feel. I think we are very good in assessing ourselves when we are stressed. People feel it, and we can discuss what it means and what you feel. But we're also very good in assessing stress in the people around us, in our family, our kids, our spouses, and our neighbors, and so on. If we see them, and the big question is why we really need those feelings. So it's clearly there's a lot of uncertainty. We worried about being infected or our loved one to be infected, but there's also a lot of other stressors which are due to the situation. Of course, some of us in isolation. Isolation is a very stressful situation, and it's probably one of the main causes for depression. We cannot travel as we used to travel. We have to spend a lot of times at home with the family members, and this definitely creating tension. We listening to the news. Israelis are very used to it. We listen to the news every 30 minutes. I don't know how it's in Canada. Again, a major source for additional stressors. So there's a lot of very unique stressor also for the current situation, and mainly the uncertainty, when it's gonna over, when we're gonna be out of this situation, and this is extremely stressful days. I would also mention this, of course, very individual. Many people both perceive the stress and respond to stress very individually. We'll not go to the reason why. It's dependent on your past experience, on your genetics, and other many factors. But the reason it's important to understand is the fact that when this response is chronic, mainly when it's chronic, and when it's not properly controlled or regulated, is strongly associated with many type of pathologies, okay, like depression and anxiety and eating disorders, but also metabolic related and heart related and immune related. But what it's important for me to emphasize is the fact that this is a totally normal and healthy response, okay? The way your brain and body is responding to this challenge, to these changes, to these stressors, is something that is aimed to protect you, okay? It's a survival response. It is something that all organisms, from fish to human, have. It's a very conserved through evolution, and it should help you to survive a challenge. Usually, unlike other challenges, if someone just, riding your bicycle and someone just cut you on the road, your brain will activate this response, but then after a few seconds, you realize everything is fine, and the system will switch off and go back to the original steady state. What we're experiencing here is a chronic situation, meaning we always stress. And again, we activate the system again and again and again. I didn't mention what it means to activate the system. So I will mention this briefly. So let's assume we hear the news, or we got just a text message and someone just told us, there is 250 people infected in your neighborhood. It's something you didn't do before. This is definitely very stressful. Or you get a message, I don't know if you have it in Canada, but we getting messages on our cell phone. You visited this supermarket on March 25th on 3 p.m. and there was someone infected there. So we have our Israeli Secret Service covering all cell phones, and we get this type of messages, and this is becoming very stressful. You get this message. What will happen in a millisecond, you receive this message, your brain will activate what we call the central stress response, okay? And it will activate your body. You will have increase in your heart rate, in your blood pressure, in your respiration, in your blood glucose level. Your sugar in the blood will go from 100 to 160 on to 180. Your cortisol, your stress hormone level will go high, but also centrally within your brain, your emotions going to change. You're going to have increase in your anxiety, in your fear. Your cognition is going to change. Your memory is going to change. Your focus attention will change. Your appetite will change. Your locomotion will change. Almost every major brain center will change its activity because of this activation. But as I mentioned before, Are you all frozen? No, it's only Jeff. Okay, I'm seeing Jeff frozen, so I will continue. So, of course, getting all the system out of homeostasis, out of control, is good for a very specific window in which you need to survive the challenge. But of course, it's not healthy to be like this for a chronic period of time, okay, for a long period of time. And this is the reason it's very important to switch off the system and go back to the initial steady state. And the big question is how to do it. How can we help our system, our brain, our body, which is, this is the way to react, and this is the normal and healthy way to react. However, it's clearly not healthy to be like this for long periods. And the big question is, what can we do? And usually I tell people, you know, one of the most relaxing things is just to interact with people. You know, you go to a bar, you get a drink, you go to party, you go to the theater, you go to the opera, and all these are not allowed today, which of course it's limiting the options. We, of course, hugging and kissing and any other warming affections are also very comforting and stress reducing, but also this we cannot do anymore, or at least it was been limited in quite dramatically. We usually can go and travel. Israelis love to travel around the world, but now we cannot even travel. And so what else can we do? And I can suggest a few things. All of you are, I'm sure, can suggest others options. One thing is what we do now is using platform like Zoom or other platforms just to stay connected and communicate. I think it's amazing. I'm doing this for more than a month now, few times a day, with our friends abroad, internally, with our employees, with my colleagues, with other scientists, with my students, and it's working very, very well. So as much as you can do that, I think it's it would be great. Other option is to do, and you don't need too much space, and you even don't need people around you, is to do any type of meditation. Those of you been experienced with, for example, mindfulness, which, as you know, as a scientist, I like to always suggesting that we have scientific evidence that is actually doing something. And with mindfulness, we do have. There's a lot of data from brain imaging and other and other methodologies that this is doing something. It's changing something in your brain, and there's different way to do it. And it's for most of the people who experience this, find it very, very helpful. We definitely should continue. Those of you who are usually talking to someone, a psychologist, a psychotherapist, many of my colleagues and friends from the field continue doing this online. And most of the psychotherapists, psychologists continue to give those sessions. I know it's different, but they do it online, and it's helpful. It's very, very helpful. A very important solution is exercise. I know we cannot go outside and run. At least in Israel, we are limited to 100 meters. So I see people running around the block or the house, but it doesn't have to be aerobic, doesn't have to be running. It could be muscle activity. You don't need to be an Ironman. Everyone, he or she, according to his or her ability, more than you usually do. And we know this scientifically. We know that is increasing the number of newborn neurons in your brain. It's very similar to what antidepressants are doing to your brain. So as much as you can do any type of activity, this is definitely going to be helpful. And of course, there's more and more things we can discuss, but I think now exactly my time is over. And sorry for rushing, but Susan, I'm very happy. Maybe I would just mention at the end, because I didn't mention in the beginning of all the effort at the Weizmann. I would just mention that we created this Corona Fund a week ago to help support. As you know, the Corona was not in our plan. It was not budgeted at the Weizmann. we do a lot of research suddenly, which is shifted toward the corona. We have all this effort. We're helping with the government. And so probably Susan will tell you more details about it. I just wanted to raise this and how important is it also to the Weizmann in these very challenging days. So it's opening for questions. Thank you very much, everyone.
Thank you so much. You know, it takes a village. And I've said before, you know, when you took on the presidency December 1st, I'm sure you had no idea what was in store for you. And you and your team are doing an outstanding job, both of protecting our scientists and our institute, and also making a tremendous impact in solving this crisis. And it takes a village. Kelly, are you still on the call? Do I see you? Yes. So Kelly and her team of getting information for us to share, videos and press releases, it really takes a village. And on the other side of the ocean, on this side of the ocean, our team, the Weizmann Canada team, developing this website and materials to share hope and share science. So I'm very grateful for everyone. And as I said earlier, most importantly, we can't make this research happen without our donors. So I do have some questions that have come to me by email. So I did put my email address in the chat, susan@weizmann.ca. So here's the first few. I have heard about studies using plasma from people who have recovered. Any comments?
It's a fantastic question. First, yeah, I mean, I would just want to say something before, which is related to this. Everyone is talking about vaccine or vaccination, and you should know that it's not always very trivial. And I know there's in the news every day is a week, there is other company or another institution that just is developing antibody. It will come in a week or 2 or 3. I remind you that other viruses like HIV or Ebola don't have vaccines for more than 30 or 40 years, although many labs try. So there is a lot of uncertainty about antibodies, vaccination. However, what just Susan mentioned is using plasma, or even more than that, using IgG or using immunoglobulin, which are isolated from these plasmas, can be used for treating people. And this is definitely something that is running now. In Israel, we have 500 people which were recovered already from the corona, have been positive and now feeling well. And those people are donating blood. They can do it every 6 days. And there is, and the idea, you can either treat immediately with the plasma or with the serum, but better is to isolate the antibodies from the blood of those people. However, this is not a vaccination, okay? This is good for probably 3 to 4 months and should mainly be used for people which are in very severe condition. We calculated this a few days ago, and I'm sitting in this government committee, and I mean, it's really depend on the dose, on the titer, but usually a person like this could be treating 2 to 3 people. So it's not medication, but of course with time, you're gonna have more and more people which are recovered. But the majority will recover. So it's definitely a good approach and should be adopted.
Thank you. The next question that came in, can you comment on about the malaria drug, hydroxychloroquine?
Okay, so the malaria drug is the first line of in the treatment currently. It's useful in some of the cases. Usually it's given with a combination of other things. It's been found useful. Also, we can learn a lot, by the way, of what has been done in China and Singapore and South Korea. We have few cases in Israel, even very severe cases that have been fully recovered. But again, this is not a treatment for corona. It has to be clear. And this is antiviral approach, so it's useful, but not for everyone. There is a new treatment drug which is also reducing the length of the disease, developed in Japan. I don't know if you noticed, but in Japan there is a lot of it's a cultural issue also, but they're controlling it very, very well. And they have a new drug that Israel received a few days ago for clinical trial. It's not a treatment, but is an antiviral which reduce dramatically the length of the disease to 4 to 5 days. Susan, I don't hear you.
It's
Oh,
actually
now.
quite easy. I have to unmute, which I forgot to do. The next question, what is the effectiveness of ultraviolet light?
Ultraviolet light. Okay, you mean as a treatment? No. Ultraviolet light is a good we use ultraviolet light in order to usually to kill bacteria and viruses as a type of sterilization, but with viruses it will not be very helpful. You need to use alcohol, not ultraviolet light. I don't think it's effective in this case. And this is just to prevent infection. I'm assuming the person was talking about killing the viruses when it's on surfaces. But 70% alcohol is killing all viruses immediately. So this is the best way to do it, okay?
Great, thank you. Next question is, what are the best possibilities and timeframes for the development of treatment and also vaccine? I think you touched upon a vaccine.
I talked about vaccine. Vaccine, it's hard to predict because of the antibody. I remind you that viruses are changing a lot. Don't forget this, okay? Viruses are mutating and creating new strains all the time. So we don't really know. We already know that there's mutation in these viruses, but we don't know yet what is the really the ability of the virus to mutate, because we are not really pushing it. And it means we're going to have a treatment, then this virus will have a pressure and you start mutating. So this is something you also need to remember. Regarding other target or molecules which will prevent the infection or prevent the proliferation, I think the chances this will come sooner is higher than vaccination. There's a lot of effort worldwide. There's many molecules now here also at the Weizmann just are being screened, and I mentioned Neil London's work. It's hard to make a prediction. When you have a molecule, it's usually easier to synthesize more and have it as a drug, especially if it's approved very fast with the FDA. Unlike vaccination, which require more time. Even if you have a good vaccination now, it will take 6 months, 1 year until it will be available in the amount that we need.
So dream forward for us, and we won't tell anyone that you said it.
Okay.
What do you think? How long do you think before we have something?
I think it will take many months. I think it will go. I think it will go away. The corona, at least, we're all gonna go back. It will not go away very fast, but we're gonna go back to our normal, almost normal life. I think in few months. It's really depend on each country and the exit strategy. I think in Israel we will go out, not in the very long term, maybe 1 month. And it will, as long as it's going to be monitored. The big fear is that we don't have more infection, everyone going to go back, and then we're going to have a second wave. The idea is to monitor it. This is the reason I talked about doing a lot of tests. And if you have a town, if you have a city, and if you have neighborhood which are heavily infected, they should not go out. So the idea is to give everyone a card, whether a green card or a red card or a yellow card, and they will be able to go to work based on their card. And this is the way we'll need to learn to work with this. We'll need to continue going with masks, with gloves, and prevent the next infection. We need to learn to live with this. And I think with time, in Israel at least, in a matter of weeks, the economy, in order to protect the economy also, we'll start releasing people to go back to work. I really hope so. And this is my optimistic, and I'm a very optimistic person. So it's my optimistic.
Alon, do you have time for 2 more questions?
I have plenty of time. It's awesome. Almost holiday here. So, awesome. And okay, I got the last meeting for today. So I'm very happy.
Yay. So we have, I have 5 questions. So now I can ask all of them. So that's awesome. So, I understood, I'm reading the question, I understood that ultraviolet light was being used to sanitize planes and buses. Did you hear about that? To the best of
my knowledge, I'm not familiar with this. I know this from my background and from the way we work with bacteria and viruses. We usually use ultraviolet, because ultraviolet is causing mutations, okay? This is the reason is is killing bacteria. With viruses, usually it's not effective, at least probably not in the same degree. Always been working with viruses. The best way to use is 70% alcohol. You see what I'm using? Look what I have here. I have this huge bucket here, which these are, each one of us got this, and these are wipes, which are 70% alcohol wipes. So everywhere I go, I do like this. I go to the elevator, I press with this. I go to the making a coffee, I'm using this, okay? And the majority of the infections are not, okay, through the breathing, okay? It's through touching, okay? The virus is not very good in spreading while I'm talking to you, or this is the reason we keep distancing, because it's coughing and sneezing, okay? But the main problem is very stable on surfaces, okay? It can even stay almost 2 weeks on some of the surfaces. So the best thing to do is always protect your hand, because you touch something and then you do like this or like this or like this, and you're being infected. So this is the solution.
Thank you. You just answered one of those questions, which is great. Do you believe and would you subscribe to the strategy of massive testing, certifying individuals and returning those who are supposedly immune back to work? These are all great questions.
Definitely, yes. Okay, this is the way to do it. Okay, this is the way we need to check people. The problem is the current gold standard for testing people is PCR. Okay, and however, the most important test to do today is serology, meaning it's antibody. We need to know who have actually antibodies. Antibodies are the indication that this person has been infected and now is protected. So, and this is a lot of efforts now worldwide to try to have a good test for detecting antibodies in the blood. But I fully agree with this strategy. People which are already infected, and I will mention again, I didn't mention, but I will mention the fact that we don't have any evidence for recurrent infection. So if someone were sick and is healthy now, recovered, it will not be infected again. There was a study done in non-human primates, tried to infect twice, and it's not working. So you can be sick currently only once, which is great. So someone who's been positive and is negative now should go back to work.
Thank you. Another question is, aside from what we've been reading, is there any advice you have of other things we should not be doing?
Not be doing. Meaning, I think we should avoid as much as possible eventually interactions, keeping distance and keeping our hands from any surfaces which are not clearly clean, okay? Or using any type, always go with this alcohol gel or any other type of wipes. I think this is the best strategy currently, because we learned a lot about the viruses, this specific virus, in the last few weeks. And I think with discipline, and it was clearly done in Asia, you know, in China and in South Korea, and it has been almost solved mainly by people listening to the instruction and keeping distancing from people. And almost all of them are back to normal already in China and South Korea. So I think, in my view, this is the solution.
Thank you. And the last question, and we're so grateful that you extended your time beyond what you originally told me, so thank you. You more or less grew up at the Weizmann. Why do you feel, or do you feel, that the Weizmann is uniquely positioned to make an impact on this COVID-19?
It's a great question. I think, knowing the Weizmann now for more than 25 years, which is half of my life since I joined the Weizmann. And I think eventually, regardless of this specific pandemic, if it's a corona, if we have other issue, maybe if we have something which relate to the universe, and we have other issues, if it's in physics, I think eventually high quality people, smart people, will come with the solution. So if you need people to find solutions, and new solutions. Eventually you need creative people, and you have this amazing creativity here on campus, and you can see how suddenly 50 groups who never did Corona research suddenly doing Corona. All the way from, I don't know, Noam Sobel now is assessing smelling coronaviruses. Other people which used to do imaging on cells are developing other techniques to do it, and antibodies and so on. So I think creativity, resources, I think equipment-wise, the Weizmann is very good equipped, and the government, for example, in Israel understood this, and we have to shift what we do, and we did it quite amazingly. Eventually, it's all about the quality of our scientists and students. And I should also mention, Susan, because this is quite amazing, the number of volunteers we have. I remind you that most of our students and postdocs and scientists cannot do their own research because we are limited for the 30% of the people. We have more than 200 students and postdocs volunteering to do the test, despite the fact they could be exposed to the virus, despite the fact that they could just stay home and watch Netflix. No, they decide to come here and work, and this is amazing. And this is across all members of the Weizmann community and family. It's technicians and people from administration, people who don't continue getting salary but they're on forced vacation at home. Everyone continue to work. And it's amazing to see this collegiality and how people are really would like to support and contribute from their time and efforts.
Thank you. And I'm sure, you know, as we talked about earlier, the multidisciplinary approach, because it's in the DNA of Weizmann, looking at the same problem from a different angle certainly helps. I got a question
I fully agree, Susan. Thank you.
I got a question by text that combines your two expertise. So one final one slipped in. Can stress make you more vulnerable to COVID-19?
Fantastic question. I got the same question yesterday, talking to our employees again. Yes, stress in general make you more vulnerable. The fact is that stress affects your immune system. And the minute it's affecting your immune system, you're more susceptible, more vulnerable to develop a disease, regardless of the type of disease. So when you reduce your stress, you actually protect yourself from any type of infection. So it's important to know.
So that's a great way to end a great conversation, because we're now going to take a few deep breaths and release any stress that we have. And you have our support on behalf of Weizmann Canada to help empower you and your scientists to move this important research forward. And so thank you, Kelly, thank you, and all of the management team from the Institute.
Susan?
Yes.
It's Lister. I only want to end with one thing.
Sure.
Kol hakavod lekol hakovid.
Thank you very much, Lester. Thank you, everyone, for taking the time. And John, thank you for the support, the generosity. And I'm going through the photos now because now I can click and see all your faces.
I just want to say you're amazing. Amazing.
Thank you very,
Fantastic
very
from
much.
Canada. We love you.
Thank you very much. Thank you, everyone.
Thank
I
you,
would
Alon.
like also to take this opportunity to wish you a happy Passover and happy Easter.
Chag Sameach.
And stay safe and well, and Chag Sameach. And thank you very much. Bye.
Toda rabah.
Bye. Chag
Bye.
Sameach.
Thank you.
Chag Sameach. Chag Sameach.
Bye.